So far in this course, we've been discussing statistical tests to assess specific null hypotheses using distributions and p-values and so forth. But this video introduces the new type of technique called ordination, and specifically we're going to talk about principal component analysis in this video. These ordination methods are types of data analysis that are designed primarily to simplify and to visualize sort of complicated multivariate data in order to aid in the interpretation of underlying processes that contribute to that data. So a lot of the data that you encounter as a scientist or a geoscientist is multivariate. There are counts or measurements of many different variables in many different sites or samples. So this type of data is difficult or really often impossible to analyze qualitatively. You can't just look at this and figure out, and we want to know like which samples are most similar to each other. Or maybe just samples from particular categories, like rock types or tectonic settings in this case, tend to group together. But also want to know which variables, like elements in this case, behave in a similar way. And so by knowing these things, we can tell something about the processes that govern the distribution of these samples and their occurrence and their abundances and so forth. So though multivariate data is complex, certainly, there is a way forward for interpreting it. And that is that there's often some degree of covariation between the variables, and this introduces redundancy into the data set. So for example, in the examples below, the lutetium value on the left covaries extremely strongly with the ytterbium value, and same with the yttrium value. So if we know ytterbium, we effectively don't need to know anything else. We don't need to measure or examine lutetium. We just kind of can pretty much guess what it's going to be because of the covariance. So ordination methods take advantage of this redundant information to reduce the number of dimensions, which allows us to visualize and interpret the patterns more easily. So the goals of ordination are basically data analysis, classification, visualization, simplifying the data to aid in interpreting multivariate data, looking for gradients or patterns. I'll explain in more detail, but basically the method essentially creates a new coordinate system by rotating the original data. This allows us to focus on a reduced number of important dimensions. And so those dimensions are synthetic variables, and they represent linear combinations of the original variable. So in this example here, axis 1 corresponds largely to changes in pH and bicarbonate. So I'll explain what these arrows mean towards the end. So the main premise is that the differences between samples, or between sites or whatever, reflect underlying processes, and those samples can be arranged along gradients in those processes. So the process itself might not be measurable or observable. This is probably especially true in the deep time rock record or a lot of the geological observations we might have. But we could try to assess that gradient indirectly from the composition of the samples themselves. So this is using ordination for something called indirect gradient analysis. So for example, if we measure the chemistry in a bunch of different lakes, we may be able to determine that an urban versus rural gradient is the most important control on their chemistry and so forth. So there are a variety of types of ordination, different strengths and weaknesses. They're applicable in different circumstances with different types of data. This table lists some of the major ones. They're grouped in two ways. First, by how they measure distance between points, and then second, by how they determine the new axes that we use for data rotation. So some methods allow you to choose the measure that you're going to use for distance. You can choose one that might be appropriate for your particular type of data, but others use a predefined and fixed distance measure. So principal component analysis uses Euclidean distance between the points, as you'll see. And PCA also determines the new axes through a process called eigenanalysis, which uses the eigenvectors of a matrix. Other methods can use a more iterative trial and error type non-parametric method. And so I'll discuss non-metric multidimensional scaling in the next video, but the other two examples listed here we're not going to cover in this class. Okay, so given that some of the variables are going to covary, perhaps quite strongly, how does PCA help us reduce the data and display the information in fewer dimensions? Well, I'll give a sort of a simple graphical illustration first and then explain in more detail how this process is achieved. So basically, PCA finds the direction of maximum variance through the multidimensional cloud of points. I'm showing a two-dimensional example here for sort of simplicity. That axis is called principal component 1. It's a linear combination of our original variables that explains the maximum amount of variance in the multivariate space. So the points are then rotated such that PC1 becomes the horizontal axis. The remaining principal component axes are orthogonal or perpendicular to all the previous principal components and are ordered by decreasing variance explained. So PC2 explains the second largest amount of variability in the original data, PC3 is the third, and so forth, depending on however many variables you have in your original data set. I'm showing you this in two dimensions. It's fairly easy to visualize the direction that's perpendicular to PC1, but it works in multidimensional space as well. Okay, so one way to describe the structure of the data, the relationship among the variables, is with this variance-covariance matrix. So it gives the variance of each measurement as the diagonals in bold in the example here, and the covariance between all the pairs of variables. So this particular matrix, like all matrices, has properties called eigenvectors. And you may remember from the previous videos that vectors can be used to describe directions in coordinate space. So I explained eigenvectors in more detail in the previous Mahalanobis distance video, so I won't go into them anymore here. It's not that important, but just remember that matrices have these things called eigenvectors. The important thing really is that the first eigenvector defines a direction in space, and the line along that direction turns out to be the direction of greatest variance through our multidimensional cloud of points. So the first eigenvector is the first principal component in our new coordinate system. So we're going to use the eigenvectors. There's going to be a second one and a third one, and however many more you have for the original number of variables. We'll use these eigenvectors as axes in our new coordinate system, and then rotate the multivariate data so that the first eigenvector becomes the first axis in PCA space. The second eigenvector is perpendicular to the first one and explains the second largest amount of variance. So it becomes principal component 2. And all subsequent eigenvectors are perpendicular to the previous ones, and each one explains successively less of the original variability. So each eigenvector, or principal component as we can call them here, has something called an eigenvalue. I explained what that was in the Mahalanobis distance video as well, but in this situation, the eigenvalue measures the amount of variance along that axis. So as I said in the previous slide, the eigenvectors are ordered in terms of decreasing variance explained. So in the example here, what you can see is that the total of the three eigenvalues, given the Greek letter lambda, is the same as the sum of the three original variances. So we can basically say that each principal component axis, each eigenvector, explains a certain proportion of the original variance, and that proportion is given by its eigenvalue. So using the eigenvectors as our new coordinate system, we can rotate the data such that principal component 1 is telling us and reflecting the most important direction of variability. In the example here, PC1 explains essentially all the variance, which is really unusual, but that's because I chose sort of an example case which had extremely unequal variances. But regardless, principal component 1 in your data will almost certainly explain less variance than this, but it still is going to represent what is probably the most important underlying process that causes this gradient of samples. So the principal component axes now are linear combinations of the original variable due to the rotation of the points. So we rotate the points into this new coordinate system. So the contribution of each original variable to a particular axis is indicated by an arrow on the plot called a loading. So the loadings are essentially the correlation between the original variable and the axis. So they indicate the direction and the magnitude in the increase in that variable. So a longer arrow indicates a stronger correlation. That means that the variable is more important in controlling or in explaining the difference between the samples along that axis. So to illustrate how that works, in this example here, I've color-coded each point by its yttrium content in the diagram. So the loading, you can see, is a fairly long arrow pointing straight to the right along axis 1, and the colors indicate, in fact, that points on the very left-hand end of axis 1 do have very small values, and points on the right-hand end of axis 1 have large values. And this is what the arrow is telling us. It says, on axis 1, from left to right, the yttrium content increases in the samples. So the example that I demonstrated used the eigenvectors of the variance-covariance matrix. But that's not the only way, and it probably wasn't the best way for the example that I used either. In fact, the choice of association matrix is a very important decision that you have to make. So the other option is to use the eigenvectors of the correlation matrix. So just to sort of compare these two, the covariance matrix centers each variable on the mean, but the scale of the variable still matters. Variables that originally had really high variance will control the resulting PCA plot much more strongly. Variables that originally had very tiny variance are going to be completely unimportant. So you should only use the covariance matrix when all of your variables are measured in comparable units, and the differences between their original variances are meaningful, and you care about that, and you want to use that for interpretation. So the correlation matrix uses a matrix of Pearson's correlation coefficients. And if you remember way back to the lecture on correlation, this has the effect of standardizing each variable so that each one has a variance of 1. So what this is best in the case when you want to remove any effect of different variances, and you would pretty much use the correlation matrix if your variables are measured in different units. Because if they're measured in different units, their variance and the differences between their variances are going to be unimportant. So PCA just rotates the axes to this new coordinate system, but there still are just as many axes as there were in the original data. If you have 10 variables, you get 10 principal components. And so the whole point, really, as I've been saying, is to simplify the data for interpretation. So obviously we can't look at all 10, but how many should we look at? So there turn out to be several rules of thumb. One is that you can make this plot here called a scree plot, which shows the proportion of variance explained by each principal component. And there's often a break point somewhere between a steep part of the line and a shallower part of the points. And you can use that break point to be sort of a cutoff. In this case, we might say, well, I'm going to look at the first 6 principal components, or something like that. You can also potentially look at every component that, when you add them up, sum up to some predetermined amount of variance explained. Like say, I'm going to look at all the components that add up to 75% of the variance or 90% of the variance. Or you could also potentially say ignore any component that explains less than 1% of the variance or less than the average variance. But in reality, people typically just look at the first 2 or 3 components. Those are the most important ones. Or you could look at however many you can interpret, right? If you don't know what principal component axis 4 is telling you, if you don't know what that gradient is, there's not a huge amount of point in spending a lot of time interpreting it. So PCA has a couple really important assumptions. First, because it uses covariance or parametric correlation, Pearson's correlation, depending on the association matrix you use, it's based on the Euclidean distance between points. So in many cases that's okay, but it might not be appropriate distance measure for certain types of data. And in particular, PCA is not a good method to use for count data. So if you have counts of the abundance of species or rock types or minerals or whatever, counts are not good for PCA generally. And second, the use of either covariance or correlation both assume that there's a linear relationship between the variables, which is again not necessarily appropriate for certain systems. Again, counts of abundance, particularly of species, is an example because those species abundances are not linearly related along these gradients generally. So if the relationships are nonlinear, PCA can produce something called a horseshoe effect, where points along axis 2 are kind of twisted into the shape of sort of a horseshoe relative to axis 1. The problem is you can never tell if your points are deformed or not because you don't know what the true pattern should be. But in any case, there's sort of certain things like abundance counts that you should not use PCA for, but the next video will discuss a more flexible method for ordination, one that specifically can be used if you have data, like abundance count, that don't meet the assumptions for principal components analysis. So that's called non-metric multidimensional scaling, and that's what the next video will focus on.